In this lecture, I'm going to explain a process called saltatory conduction. And I mentioned the fact that neurons have this coating called the myelin sheath, and I referred to it as kind of like an insulation. And I mentioned the fact that it allows the impulse to travel down the axon. And I also mentioned these spaces, these gaps in the myelin called the nodes of Ranvier, and I said this is how the neuron is able to send the impulse so quickly. Well, what I'm going to do today is I'm going to explain why. Why is that the case? So let's get started. So what is saltatory conduction? Well, saltatory conduction has nothing to do with salt. Saltatory actually means to leap. It's Latin. It means to leap. And that is where they got this from. And what ends up happening is the impulse, as it travels down the neuron, it does so because it jumps over these little gaps in this myelin sheath. So the myelin coating is made of fats. It's mostly fats, and it wraps around the neurons. And just as a reminder, there are two types of cells that produce the myelin sheath. In the peripheral nervous system, so everything outside of this, the spinal cord and the brain, there is a cell that is called a Schwann cell. And that Schwann cell tightly wraps around the axon, which is what creates this myelin sheath. And then in the central nervous system, there are these glial cells called oligodendrocytes, and they do the same thing, but you'll have one oligodendrocyte that wraps itself around several neurons, and that's what helps kind of keep these bundles of neurons together, but it also produces this myelin sheath. So here's the thing though, not all neurons are myelinated. Some have myelin, some don't. And you can actually see the difference. So we're going to talk about this when we get to the brain unit, but you may have heard of the gray matter and the white matter of the brain. Well, the reason why they're called the gray and white matter is because that's what color they are when you preserve it. So when you guys like dissect the brain, which we hopefully get to, when you cut the brain open, you'll see that there's a distinct difference in color between the inner portion of the brain, which is the white matter, and the outer portion of the brain, which is the gray matter. And the difference is myelin. Fat is white. So if you've ever cut meat off of a steak, fat is white in color. Well, the same thing is true with myelin. So the area that is white matter is actually, that's going to be myelinated axons. And the area that's gray matter is going to be unmyelinated. So why do we have two types? Well, the reason why is because some axons are really long. And because they're long, they have to send information a distance very quickly. So we have this stuff called myelin. And it works by essentially insulating the neuron and preventing any passage of ion flow where the myelin is, and then there are these gaps where that can occur. So I'm going to explain this better. Don't worry. So just to kind of finish up this slide here, the channels are essentially, they're only going to be located where the nodes are. So if you take a look here, this shows you, actually this is an actual node. This is from an electron micrograph here. You can see there's the myelin kind of coating the axon, and then there's this gap area where the node is. That's what corresponds to here, and this is where all those channels are. And I'll explain that better in a minute. And then finally on the slide I have myelin, which is going to be white matter, and that's going to be faster than the unmyelinated. So I'm going to explain this with a little, surprise, a little analogy. If you've ever gone down to Philly for a game, if you've ever gone to the stadiums, you probably took the train before. So the way the city's set up is pretty simple. So you have City Hall, right, which is right here. So that's City Hall. And then the stadiums are down here in South Philly. And if you go to Philly, you might take the train to go to a game. Makes sense, right? So you take the train in and you get off, and then at City Hall, you can jump on the orange line. And the orange line just goes straight down Broad Street to the stadiums. But there's going to be two options for that. There's going to be the express train and the local train. And you want to get on the express train, and here's why. The express train only stops at the major stations, along the way. So maybe it stops at, 6 or 7 points along the trail because it's the express train. The local train, on the other hand, stops at every single station on the way down because people have to get on and off. It's the local people. They live here. And then on the express train, these are the people that are just trying to get to the stadium the fastest. So if two trains leave this point at the same time, obviously the express train is going to get there first because it's going to go skip a whole bunch of stops, right? It's going to jump from major station to major station. The local train, on the other hand, has to stop at every station along the way. Even if you don't have to get off at those stops, it still has to make those stops along the way. So two trains that are going the same speed initially are going to eventually start to spread where the express train is going to go much faster. And that is exactly how this process works. So let me explain it real quick. So up here I have two diagrams I drew. In the first diagram, I have a myelinated axon. And as you can see, we have our nodes of Ranvier, and this is going to be where we have our channels. Let's say the blue blocks represent sodium channels, and the yellow blocks represent potassium channels. And then down here, we have what would be an unmyelinated axon, where just all along the entire membrane, we have these stations, essentially, these sodium and potassium channels. So let's say we have an action potential. So I have an action potential that occurs, right? And that sends an impulse. Sodium flows in, potassium flows out. Well, what's going to happen here is the sodium that flows in from this action potential is going to be what triggers, because this is insulated, there's no loss of charge. So this is able to keep that charge going, and it's going to move all the way to here, and that's going to trigger this channel to allow sodium in. And then you're going to have another action potential, and then the sodium from this action potential is going to trigger the next one. So essentially it becomes a scenario where one action potential triggers the next, but because there's this big gap here where there's no exchange of ions, this very quickly jumps to the next point, node. Down here, if this is an unmyelinated axon, you're going to have the same thing occurring, but because these are all so closely spaced, you're going to have an action potential happen here, and then it's going to happen here, and this is going to go all the way down the line, just like it's supposed to, but obviously it's going to take longer. So I actually have a good little animation here, which I want to share with you. All right, so this is a little animation of this process. Let me move myself here. So I'm going to play it. It's really short. So both get stimulated, and as you can see, up here it's jumping down. So they start at the same point. Look at the difference in conduction velocity. It's going to be a massive difference where, and here it zooms in, you can see it skips under the channel. Those positive charges trigger the next one, and it moves down the line. So I'm going to finish up here with what happens when the myelin gets worn away. So there's a lot of diseases, one being multiple sclerosis. You may have heard of multiple sclerosis before. It's a devastating disorder where the body actually attacks itself. It's an immune disorder, and what ends up happening is it actually attacks the myelin. So the myelin sheath gets removed from the neurons. There's another disorder called adrenoleukodystrophy, or ALD, and we are going to hopefully be watching a movie in here called Lorenzo's Oil. It's a true story about a treatment that was developed by parents of a child that had this devastating disease. But ALD is a condition where actually what ends up happening is the myelin gets worn away, and what ends up happening is the neurons stop working and the person loses motor function, and eventually obviously they can't move anymore, and eventually their breathing stops because their diaphragm, they have to be on a respirator, and it's a devastating disease. So why would that happen? Why wouldn't it just work without the myelin? Well, let's imagine it this way. There are no channels here, right? The cell's not going to produce channels where they don't need to be. Imagine putting windows in a wall that went up against another wall. There's no reason to put a window in because it's a waste of money and energy and everything else. So the cell doesn't put protein channels where the myelin is. So let's say there was an instance where that myelin gets worn away, right? Multiple sclerosis or ALD. What ends up happening is this neuron doesn't work anymore because now this gap here where that myelin was, it used to insulate it. So this impulse was able to make the entire trip. But now because that gap doesn't exist anymore, now there's no longer that myelin, there'll be, it'll lose its intensity. It'll lose, that sodium will just diffuse out on its own because there are some sodium leakage channels, and essentially it won't make the trip. So you can't just wear it away and be okay. And that's why those diseases work that way. Thank you very much.